So I'm Filipa Godoy. I'm an associate professor at the Department of Microbiology and Medical Zoology at the University of Puerto Rico School of Medicine. I'm also the chair of the department, and I'm directing the microbiome lab that started about 3 years ago. I moved from another institution here in Puerto Rico, where I developed the first human and animal microbiome projects here in Puerto Rico. So how did I end up in bioinformatics? I'm actually not in bioinformatics. I use bioinformatics as a tool. I'm an ecologist by training. I did my bachelor's in Portugal. I'm Portuguese. I did my bachelor's on plant cell walls, and then I did a specialization in algae. And when I came to Puerto Rico, I wanted to do something in tropical ecology. And I ended up working with a Hoatzin. I was working under Dr. Dominguez Bello, one of the pioneers of the microbiome field. And we started several studies on different animal microbiomes. And my focus was on pre-gastric fermentation. So I studied bacteria and fungi that are part of the gut microbiota of the only folivorous bird. It's called the flying cow. So I studied the Hoatzin microbial ecology through my Ph.D. Then after an NSF postdoctoral fellowship, I worked at the DOE Joint Genome Institute, where I learned metagenomics. So I always use bioinformatics as a tool. I don't see myself as a bioinformatician or a programmer. I do adapt programs from other people or pipelines that are already available. But to study host-microbe interactions. But actually, I started learning by myself. It was because about 15 years ago, there was no universal pipelines for studying these things. We sequenced with Sanger, and we picked colonies and sequenced white colonies in order to figure out if our amplicon was in this bacteria. So we did cloning and transformation. I'm still from that time. And so we had longer reads, and the phylogenetic trees would take like 1 or 2 weeks to finish. Basically, I learned from other people that were more knowledgeable of this bioinformatics and statistical tools. And then actually one summer I went to I'm from the time that we used to do cloning and transformation and picked up the white colonies. And we have thousands of reads from Sanger, which we wanted to do, phylogenetic trees. And I learned about this ARB package. It was one of the first Unix pipelines for microbiota, which was not a field yet, actually. So I went to Germany to the Max Planck in Bremen. They developed this package called ARB. They are now the developers of Silva, the Silva database for microbiota analysis. So I learned with them about ARB and about Unix programming, and I started getting engaged into bioinformatics. So I actually started by learning a lot from papers, from emailing experts. In fact, the PI from my postdoc was Phil Jugenholz. He's one of the creators of the Green Genes database. And I read a lot of his papers, and I was so excited to invite him to be part of my project. So I started, learning from other people and getting information from my own research. I kind of developed with the microbiome field, because when I finished my PhD was in 2008, and it was when the microbiome field really kicked off. Yeah, well, if I was a student today, that would be wonderful, because there are so many pipelines and tutorials, and everything is, digested already so that you can apply those tools. And in my time, we didn't have anything like that. We didn't have resources. We didn't have, Chime2 or, these are packages that allow the integration of so many different types of data, and it's all explained in the tutorials. In my time, we didn't have anything. So if I had to give an advice to someone, and sometimes if you're a biologist, you don't have much information on how to start using these tools, you get frustrated. For example, in R, the package doesn't read the file, and students like spend hours trying to figure out and go to the forums. And my big advice is don't get frustrated. So be resourceful and go find someone who can explain that to you. Go find a forum, subscribe to all the forums for all the pipelines that exist. Everybody explain. Nowadays, people are really generous, and at least in microbiome, the data is public and the tools are public. People don't make money out of it. They want to share their science. We want a field that evolves with knowledge, and everything is free. So you can email the experts, you can go to the forums, but never get frustrated, because it's easy to get frustrated. But nowadays the resources are out there. So go find a mentor who is also a sponsor. That I always emphasize to my students, that mentors have to be sponsors. So you have to find someone who speaks good of you when you're not in the room, right? That's a good sponsor. A good mentor is one that supports you and speaks highly about you when you're in the room. But when you're not in the room and they want to be referees for you, you want them to speak good about you. So you want sponsors, right? And there's a lot of people in different areas that can help you. So be resourceful, don't get frustrated, and solutions, and be resourceful. That's a very interesting question. Sometimes I think about it on how would I like the field to move forward, and how could I be useful to help students move forward. So in my lab, for example, we have several tutorials that have been developed through time. I started my own tutorials that have been modified and improved by my own students through time. And these are available for everyone who comes and rotates in our lab. We have sessions in the lab where students get together and answer questions and doubts that anyone has. And we have a Google Drive where we have this dry lab analysis and tutorials. And I think that empowering students, those that have more experience and they have capacity to teach other students, is a good way to do it, because sometimes the PIs are really engaged in other things, or teaching, or getting grants out, or publishing papers, and we lose touch a little bit with our students. So I always try to find some time to discuss in one-on-one, and then we have the big group meeting once a week, but we have also individual meetings. But then empowering students to help each other, I think it's a good principle. To empower your own students to be able to teach other students, create a network, and that's what we've been doing in Puerto Rico through the IMBRE Corps. So I'm also participating as a part of the BIRC, the Bioinformatics Research Corps from Puerto Rico, IMBRE. And one of the things that we do is we do a lot of workshops on bioinformatics, and we collaborate with clinicians and PIs that bring their own projects, and we think about ways that they could integrate bioinformatics, or they could integrate the microbiome. And I always involve a student from my lab to help them analyze the data and be like a bridge between us and the other groups, so that my own students can be also mentors from other labs, students from other labs. And in the end, tomorrow you'll see the talk of one of the students that I had since high school, and he's already published 3 papers. And by empowering students, the only way that can grow, and they can help you grow.
Yeah.
So I think that would be my advice to especially for mentors, to empower their students.
Yeah.
Because they're going to be the future of what we do. And if they know what they're doing, if you are sure that what they're doing is good, if they do good quality controls, if they know that how to manage the code and find solutions, you should empower them, invite them to give a talk, invite them to be part of a project that you're collaborating, that it's not their main project, but it could be a side project for them to have another publication. So empowering students, I think, is very beneficial for the field of bioinformatics. Yeah. So my lab is organized in two ways. We have a more clinical part where we specialize more in microbiome-driven malignancies. So we studied cervical cancer and solid tumors and how the microbiome can drive these phenotypes, these cancer phenotypes. We work with women. We have a big project on human papillomavirus infections in Hispanics. The rates of cervical cancer in Puerto Rico are higher than anywhere in the US in the last 10 years, despite of vaccination. So we hypothesize that a lot of the microbiome, cervicovaginal microbiome, is driving these changes, and we are studying that. And that is also part of that project is a PhD thesis and other small projects deriving from this project. Other students are involved, including medical students. And that's something that I also envision. I envision a future where medical doctors can look at microbiome data, interpret the data, and follow up with their patients, longitudinally, and they can make clinical solutions based on the microbiome. So we are also teaching MD students, and they're coming to the lab and they work with undergraduates, with graduates, and it's really an interesting and heterogeneous group. So that would be, that's something that we do. It's the part of the clinical project. And then we have the environmental projects where we have collaborations with folks studying climate change and how invasive species have different microbiomes that made them more able to colonize corals and kill them. And we're studying that seagrass microbiome. That's also for a computer science student that we have in the lab. He's just starting right now and he's learning from these other undergraduates and graduate students. So as you see, it's a big network with different projects, with different hypotheses, but with the same tools. So it's a big group that are a good net of support for each other. So besides the seagrass, we have also the Moby microbiome. The Moby is a fermented drink in the Caribbean. And it's one of the side projects from this undergraduate student that I was mentioning. And he found out that there's at least 17 different species of lactobacillus in this fermented drink. We are about to publish that. And we also have projects on turtles, marine turtles, crabs and mangroves and the differences in their niches. Ones are in the red mangrove, others in the black mangrove. So their microbial communities are derived about the different nutritory niches where they fit. We also have a sea urchin project that we just published a couple of months ago. The first study on sea urchins in Puerto Rico. So I always try to involve students and make them excited about the project. And that's another thing. If you work with bioinformatics, you have to be excited and you have to be, looking forward to seeing the results. And that should permeate from the mentor to the students. And everybody comes engaged and happy and looking forward to the results. And that's, the excitement. I think that's what drives me and my students. Oh, definitely. Yeah. And that's my dream. Like, I can imagine what you're doing, that we can involve other PIs that are interested in similar things as me. And we have sequencing facilities within the IDEA networks. We have multi-omic facilities, GC mass specs and all that. And we have a lot of bioinformaticians that could help different PIs from these IDEA states to integrate their data and do meta-analysis. We could have a summer just on meta-analysis. That we could do a workshop on, let's look at the microbiome in women, in obese, Hispanics, African Americans, Caucasians. Let's do the microbiome within these IDEA states and look at, the different cardiovascular phenotypes and possible cancer outcomes. And we could do a project on that. And we could have like a PI co-authored by students and folks in the IDEA networks. Of course, I think that's the future. We should integrate, and we are all using the same methods. We're using the same, protocols, the human microbiome and the earth microbiome protocols. So we're all using the same kits, the same everything, right? We're using the same sequencing machines. So we can use, a repository like Cheetah, which accepts, and normalizes all these data sets and integrates them. And we can definitely do meta-analysis and work on different things, not only on clinical things, but also on environmental problems that affect everyone.